Welcome back to our GlycoNet series. So today I have a great pleasure of introducing Dr. John Hanover, who is currently at the National Institutes of Health in the US. John got his PhD at Johns Hopkins University studying glycoprotein assembly, and then did a Jane Coffin-Childs postdoc at the NIH, where he got into some signal transduction and disease. So it's really exciting to bring John here because he's one of the co-discoverers and really has been one of the prime movers of the O-GlcNAc field for many years until the field started taking off. And he's just been a fantastic mentor to so many O-GlcNAc scientists as well as other glycobiologists. And so it really is a pleasure to introduce him. So John, take it away.
Thank you, Charlie. I'm going to focus today on some of the biological aspects of the O-GlcNAc modification, but I wanted to introduce the topic fairly broadly. And the reason we're interested in O-GlcNAc is it plays important roles in human physiology and is very clearly associated with human pathophysiology. It's a fairly straightforward pathway, unlike the assembly of N-glycans, which require many enzymes. There's really just two enzymes, OGT and O-GlcNAc ACE, which mediate this pathway. Environmental nutrient information is integrated through metabolism to create UDP and acetylglucosamine. This is then is the substrate by which OGT modifies its many partners. And we'll be talking about those partners today and the effects of the modification on the partners. Okay, so the evidence that O-GlcNAc's involved in disease is really quite compelling at this point. Both enzymes are essential for mouse development. There's no loss of OGT function in over 30,000 human genomes that have been sequenced, and we'll be talking about that later. It's one of 23 disease-associated genes on the X chromosome. This is by inference. OGT is X-linked imprinted, we've shown, in Turner syndrome, in which individuals inherit a single X. And OGT is induced by placental stress. Work in our lab and that of Tracy Fehl has shown that OGT expression is different in male and female fetuses. And this is due to changes in the placenta. So the placenta in which a male baby resides is actually quite different transcriptionally and is subject to placental stress. And hopefully we'll be able to talk a little more about that later. A similar skewing in gender with regard to OGT is seen in lupus, where OGT is elevated principally in females with lupus because of skewed or partial X inactivation. OGA is a diabetes and obesity susceptibility locus, particularly in the Mexican American population. OGT and OGA are both elevated in cancer. We'll discuss this in the talk. OGT is mutated in a rare intellectual disability disorder called XLID. Brain OGT and OGA null mutants induce an obesity phenotype that's really rather striking. And finally, the activity of these enzymes is linked to Alzheimer's, Parkinson, and other neurodegenerative disorders. So by way of introduction, UDP and acetylglucosamine, which is synthesized via nutrient inputs from these nutrients, glucose, glutamine, acetyl-CoA, uridine, and ATP from macronutrient breakdown, lead to the increases in UDP and acetylglucosamine, a highly regulated intermediary metabolite. UDP-GlcNAc then is the substrate for OGT, which transfers O-GlcNAc onto serine and threonine residues of many targets. Many of your favorite targets are listed here, including kinases, nuclear pores, and mitochondrial complexes. There's also a complex interplay between O-GlcNAc and phosphate. And we'll talk more about that interplay in a few minutes, but it is complicated further by the fact that over 550 kinases act at the same sites as O-GlcNAc, and many of those kinases are O-GlcNAc modified. So you can imagine the very complex interaction that occurs between these two cytosolic post-translational modifications. So we've been involved fairly heavily in the structure of OGT work in our lab, and Susanna Walker's have resulted in a complete analysis of really a pretty complete understanding of the OGT structure. We also know that O-GlcNAc transferase is ribosome associated. It's recruited by PIP3 and other inositol phosphates. It has a kinase binding domain near the catalytic surface. It has a TPR domain, or tetratricopeptide repeat domain, that provides scaffolding for interactions. It's also subject to numerous post-translational modifications. Excuse me. Our lab showed early on that the OGT gene is subject to alternative splicing, resulting in different intracellular targeting to the nucleus cytosol, to mitochondria, and within the mitochondria in the case of the short OGT isoform. So this modification, quite literally, is everywhere intracellularly. I mentioned the mitochondrial targeting because recent work has shown that the nuclear mitochondrial interactions mediated by OGT are critical to its function. It plays a role in biogenesis through the SYN3 transcription complex, apoptosis, nuclear mitochondrial fission and fusion, and we're actively working on this problem, cancer metabolism, and trafficking of mitochondria along microtubules through the so-called Milton microtubule-associated complex. So if you look at the proteins that are modified by O-GlcNAc, and we've done this somewhat systematically, they, like phosphorylation, tend to occur at intrinsically disordered domains of proteins. And some years ago, we pointed out that OGT shares similarities with important alpha, with this asparagine ladder motif that allows binding to numerous polypeptides, somewhat independent of sequence, owing to the ability to form multiple hydrogen bonds. And so several groups now have shown that this appears to be a critical component in substrate recognition. It turns out that about 98% of nuclear proteins have intrinsically disordered domains. So this is a non-trivial substrate collection. Such favorite as the nuclear pore complex, the RNA polymerase II, and the so-called polycomb bodies, all are intrinsically disordered in part of their domain. The O-GlcNAcase is no less interesting. It's actually also alternatively spliced to produce two isoforms. One of these is associated with a histone acetyl transferase domain, allowing it to enter the nucleus and reside in the cytosol. There's another isoform that lacks that hat domain, and this is targeted to lipid droplets and maybe a mechanism by which the O-GlcNAcase enters and regulates mitochondrial metabolism. So we've proposed what we call the O-GlcNAc timer hypothesis, and this is very similar to the ER mannose timer that operates in the endoplasmic reticulum, by which a specific addition and removal kinetic leads to stabilization or destabilization of a protein. And so with that in mind, I'm going to talk about some specific examples of how O-GlcNAc may mediate those functions. So how are these diverse targets recognized? So we've been carrying out cryo-EM of these enzymes so that we can look at those complexes. I should say, by way of mechanism, OGT exhibits characteristics of an ordered bye-bye mechanism. In simplified terms, that means a UDP-GlcNAc has to bind prior to the peptide, and that's part of the catalytic mechanism. These enzymes also show so-called distributive rather than processive mechanisms. That is, they operate in a distributed manner across a protein, not from one N-terminus to C-terminus, for example, no processive mechanisms. This asparagine ladder I mentioned plays a key role in binding to substrates, we think, through the TPR domain. Much less is known about O-GlcNAcase and its interactions. But we've recently solved the cryo-EM structure of both OGT and at a higher resolution, the O-GlcNAcase. So we're very hopeful that cryo-EM will be a way of understanding how complexes form and how the many and diverse substrates of OGT are recognized. Okay, getting into the main topic of today's talk, I wanted to show you a little bit about the disease targets. And this is a very small list of targets that are associated with metabolic disease, neurodegeneration, cancer, and stem cell biology. But I'll have some more specific comments about these substrates. We're currently working on galectin-3, which is an O-GlcNAc modified protein that binds surface in glycans. And so we think there'll be an integration between O-GlcNAc and the N-link glycosylation pathway. So by way of introducing what O-GlcNAc does, I wanted to just mention a few key examples of how it operates. And so the citation is here if you want to look further, but we know that O-GlcNAc can alter the activity and signaling of signaling cascades. It can alter the stability of proteins, particularly those associated with neurodegeneration. It can impact nuclear trafficking and transport, and exhibits a very complex interplay with many other post-translational modifications, including phosphate methylation and polyubiquination. So when we think about O-GlcNAc associated diseases and modeling them, these are the human diseases, lupus that we talked about, meningioma, type 2 diabetes. How do you model that with mouse or with simple model systems? And this has become even more important now that we know that a form of X-linked intellectual disability in man, so-called XLID, X-linked intellectual disability, is associated with mutations in O-GlcNAc transferase at many sites, not only the substrate binding TPRs, but 2 sites in the catalytic domain. And so it becomes really critical to understand how such hypomorphic mutations may lead to disease as extreme as X-linked intellectual disability. So one of the ways we understand this process is by looking at multiple model organisms. In human and mouse and in higher vertebrates, both OGT and O-GlcNAcase are essential. However, in simpler organisms, a highly conserved pathway in Drosophila and C. elegans allows us to do genetics, because in these organisms, O-GlcNAc is interestingly not essential. So we've used these model systems to look genetically, and this, genetics provides a wonderful way of surveying the ground. What we've learned from these models is that O-GlcNAc is associated with polycomb and Hox gene expression, playing roles in developmental plasticity and neurogenesis, in innate immunity, and particularly the stress response, in insulin-like signaling, in various forms of diapause, including torpor in vertebrates, neurodegeneration and associated autophagic degradation, macronutrient storage, stem cell biology, and I'll talk quite a bit more about this, and fertility, where male C. elegans nematodes are in fact infertile. So with that as a starting off point, I'm going to summarize some recent data which highlight the role of O-GlcNAc in stem cell and cancer epigenetics, and then talk about a number of other topics that relate to human disease. So we've recently shown that O-GlcNAc cycling controls stem cell renewal and DNA repair. And this work really came out of work in the fly, in which we looked at the so-called stem cells associated with the intestine and the gut. And the gut in the fly is regenerated very rapidly, allowing you to monitor these GFP-containing stem cells and identifying them, and using that transcriptional system to regulate such genes as O-GlcNAc and other reporter genes. So what we found was that when we knocked out the O-GlcNAcase in these fly guts, we got this tremendous dysplasia. That was also true if we carried out RNAi in these tissues. So this led to a massive stem cell hyperproliferation phenotype. Looking at its root causes, we found that it was due to the activation of DNA damage control. H2A V Drosophila, which is the H2AX homolog, incorporated into those stem cells at a very high degree. ATM, ATR, canonic kinases associated with DNA damage were activated, and CHK1 was shown to both stabilize and modify and stabilize OGT. So this leads to a kind of a vicious circle feedback loop in the stem cells, driving DNA damage and driving DNA damage repair through inactivation of this simple homeostatic regulator. And we think there's a feedback feed-forward loop regulating this. So we showed that this pathway was also conserved in mouse embryonic stem cells and MEFs. When we looked at a specific lineage in the mouse, a popular one to examine, which is the hematopoietic stem cell lineage, OGA had a very similar phenotype. That is, we got complete loss of hematopoietic stem cell and downstream progenitors. There was a complete loss of competitive repopulation in bone marrow transplant studies. There was a doubling of apoptotic cells, suggesting that this proliferation was inhibited and apoptosis was activated. And there were evidence for deregulation of genes involved in hematopoietic stem cell maintenance and specification. So let's look at the other epigenetic mechanisms that have been invoked for O-GlcNAc. I'm going to go through this very quickly. They're mostly pretty pictures of Drosophila polytene chromosomes, but we've done all of this with transcriptomics and ChIP-seq and other analyses. We find that O-GlcNAc sites are distinct from the position of elongating pol2. They often overlap with polycomb repressive complex on chromosomes. They dynamically cycle on proteins of the trithorax family class. And this is kind of a fun example of what you can do with the OGA and OGT nulls. And in the fly, OGT is called supersexcomb, because fly, you have to have a sexy name to get popular in the Drosophila community. OGT is called supersexcomb. It's essential for polycomb repression. And polycomb repression is very important for regulation of the Hox genes. Perhaps the most famous of these are the antennapedia A, antennapedia B locus. And as you can see, polycomb group and O-GlcNAc are almost exactly coincident. So in the fly, there's about 1,000 promoters like this. Most of the Hox genes have O-GlcNAc. There's no O-GlcNAc turnover at those Hox genes. In C. elegans, we have a similar situation where we have about 1,000 of these promoters. We know that OGT plays a role in cellular plasticity because loss of function mutations lose their ability to maintain cell identity. And we also were able to show a genome-wide perturbation in RNA pol2 dynamics with loss of either of these two enzymes. So the situation becomes a little more complex in the mouse. There are about 5,000 of these sites in mice and humans. They affect many transcription factors, genes associated with immunity, mitochondrial function, stress. They're actually O-GlcNAc cycling and nutrient dependent. So the size of these promoter peaks changes with nutrients. We see about a 95% overlap with CPG islands and TET binding sites, critical for reading the DNA methylation footprint. So we also know that OGT is required for DNA methylation dependent retrotransposon silencing. Some huge percentage of the human genome has to be regulated by retrotransposon silencing, particularly in the germline. And we know that RNA polymerase is a prime target. So to put all this together in a form that's hopefully a little more comprehensible, I've shown you that oncometabolites, metabolic reprogramming leads to changes in hexosamine's biosynthetic pathway, leading to changes, typically elevation in UDP-GlcNAc. OGT has numerous targets, which are listed here below, including promoter methylation and demethylation. There's also activation of the DNA damage repair pathway, leading to incorporation of H2X and glycosylation of other histones. There's the trithorax activation complex, the polycomb repressive complex, activity at the Pol2 C-terminal domain, and then finally alternative splicing via intron retention that regulates a large number of genes, including these two genes, OGA and OGT themselves. So this is a very complicated epigenetic regulation triggered by oncometabolites and UDP and acetylglucosamine. So quickly, I wanted to talk about metabolic homeostasis. It turns out that we've made transgenic animals which either overexpress OGT or are heterozygous at the haploinsufficient at the OGA locus. And these in general show metabolic phenotypes. They have increased fat mass. They have what's known as a caloric memory defect. They snack when they should be eating, and they eat when they should be sleeping. We're currently studying this in collaboration with Oleg and others. And I wanted to talk a little bit more, though, about the complete knockout, because the ability to take a flox allele, in this case, the O-GlcNAc allele, and completely knock it out allows you to develop tools such as transform mouse embryo fibroblasts, which we've used and we've made available to the community. We also look at these OGA conditional knockouts in the context of human disease. They exhibit massive obesity, patterning and skeletal transformations, hematopoietic stem cell deficits, spermatogenesis defects, and neuronal migration defects. But most importantly, they're about 95% perinatal lethal. They don't make it beyond the perinatal period. So we've looked into this using omics analysis, transcriptomics, and metabolomics. And a pathway that jumped out at us was the so-called PPAR alpha and gamma axis in the heart and liver of these knockouts. OGA expression itself is regulated by PPARs. There's a DNA methylation dependent expression of a fibroblast growth factor, FGF21, that we see abolished in the OGA knockouts. And finally, we know that FGF21 programs metabolism for offspring into adulthood. It's a very interesting molecule. It provides the so-called sweet tooth, the longing for sweets that many of us have. It is cardioprotective in protecting the heart from ischemic damage. It's regulated by a TET2-dependent demethylation reaction associated with PPAR alpha and PPAR gamma. And so we're very excited about this pathway is playing a key and perhaps causative role in metabolic homeostasis in man. We've studied the brain a little more extensively, and I'm going to end with this work on the brain. We found that knocking out OGA specifically in the brain selectively impacts neurogenesis. This is the subventricular zone, and you can see the proliferation by O-GlcNAc. This is an O-GlcNAc specific antibody in the wild type and in the OGA knockout. This is brain specific. You don't see the knockout occurring in the liver or other organs, but you do in the brain. These animals exhibit a clasping phenotype suggestive of neurodegeneration, hypotonia, a very shortened face. You can actually spot these from their litter mates. They have a very high body fat, 40% as a young mouse, and that's very rare. Even adult mice at 40% body fat are very rare. There's increased insulin, leptin, and adiponectin, suggesting changes in fat metabolism. When we looked at the brain, we found something very interesting, and that is there was a developmental delay phenotype associated with these paraventricular regions. There was also near complete absence of the pituitary in these animals. Subsequent analysis, we found that both cortical development and pituitary hormone release are dramatically delayed in these OGA knockout animals. We think due to the destabilization of OTX2, a homeobox protein associated with the forebrain in mice. There's also altered neurogenesis, and we've exploited this model as a model for both for looking at the role of O-GlcNAc in neurodegenerative disease. So by way of introduction here, neurodegenerative diseases are being studied extensively in the O-GlcNAc arena. There are many forms of neurodegenerative diseases, but they are associated with accumulation proteins intracellularly. We developed a C. elegans model that showed that blocking the cycling of O-GlcNAc was protective. Jamie Marth produced an OGT knockout in the brain and showed that tauopathy was affected by loss of OGT. There are these neurogenesis defects I've talked about, a meta-analysis suggesting that O-GlcNAc is a key deregulated metabolite in the brain. And finally, in tauopathy, there have been some protective effects observed with O-GlcNAc inhibitors. And this is work done by Merck and Eli Lilly and spearheaded by David Bacodlo. So I want to talk about our efforts to carry out some diagnostics for the role of O-GlcNAc in Alzheimer's disease and other neurodegenerative disorders. We've exploited the vast array of O-GlcNAc inhibitors that we and others have developed to develop a druggable or at least a biocompatible ligand that would be useful for imaging. This compound, which is called OGA1, was the prime candidate. We did this jointly with Eli Lilly and in collaboration with Victor Pike and Bob Ennis at the NIH. So this ligand has an IC50 of about 1.29 nanomolar, fairly decent IC50 with regard to O-GlcNAc activity. It has an M-log-D of about 2.9, so it has the characteristics of a potential ligand. It's not a substrate for P-glycoprotein or breast cancer-related protein, and it's likely to cross both the blood-brain and blood-testis barrier. So we were able to use the knockout mouse to validate this, and this is a real luxury when you develop PET ligands because you can basically rule out non-specific interactions. This is the F18 inhibitor. We've made a number of other isotope derivatives, but the F18 inhibitor is very sensitive, and it turns out to be an excellent radioligand in mouse and monkey. Here's the mouse in which you can see the knockout is basically the signal is completely gone in the knockout, and this is an MRI just to show you the level at which this imaging was occurring. In the rhesus monkey, you get a lot more detail about the localization of the ligand at the baseline. Here we carry out not genetic ablation, but blocking with a unlabeled inhibitor, and this is the MRI at that level, looking at the rhesus brain. Just to give you a sense of how this ligand is distributed in the brain, I want to show you this movie. This is the rhesus forebrain, and you can see the O-GlcNAc here in red is associated with those very structures that are associated with neurodegenerative loss in Alzheimer's disease. And so O-GlcNAc distribution is certainly consistent with a role in neurodegenerative disease. We also hope to use this ligand to look for sites of plaque formation where the levels of O-GlcNAc are likely to be much higher. So this ligand is currently in clinical safety trial at the NIH. We're hoping that such O-GlcNAc inhibitors can be used for therapeutics for tauopathies and Alzheimer's disease. We're able to see whole body distribution of O-GlcNAc, and for those who are interested, I can show you that. And so we're hopeful that these PET ligands will be very useful for Alzheimer's disease detection. So I'm going to summarize to leave plenty of time for questions. What I've told you today is that the O-GlcNAc modification occurs at domains of intrinsic disorder, influencing protein stability and folding. It plays a conserved role in DNA methylation, epigenetics, and the regulation of Hox gene expression, the very determinants of the vertebrate body plan. It's a metabolic regulator of stem cell biology and DNA damage repair. And here I think it's really important to point out that many of us in the field believe that O-GlcNAc is a key component driving the so-called Warburg effect, the elevation of the loss of oxidative phosphorylation and the increase in such pathways as pyruvate production, and which is known to drive the cancer phenotype. What's even more interesting is this DNA damage repair pathway actually calls into question whether these metabolic changes may be actually causative, leading to increased DNA damage in tumors and leading to an increase or elevation or escalation in the tumor phenotype. We've shown that PPAR FGF21 signaling is a key player in the progression to metabolic disease and obesity, and that it may be pre-programmed in the perinatal or neonatal environment. Really interesting future work will be required to determine, just what effects there are on the developing fetus due to changes in placental metabolism. And finally, I think I've shown you that O-GlcNAc is a promising target for neurodegenerative disease diagnosis and therapy. And so we continue to develop tools, chemical and pharmacologic, that will allow us to determine the role of this pathway in human disease as we can model it in model organisms. And with that, I'll stop and answer any questions you guys have. Sorry to rush through it, but we can go back to anything that I made unclear.